Great. Well, it's good. It's 7:33 on a Friday morning in November. Hard to believe. So welcome everyone to our first in-person province-wide rounds or hybrid province-wide rounds. I want to acknowledge that this is a collaborative effort by BC Renal and UBC Division of Nephrology and all of our corporate and health authority partners to ensure that we promote education and help ensure that we are as up to date as we can possibly be on everything kidney to serve our patients better. I also want to acknowledge that we're on the unceded territory of the Musqueam, Squamish, and Tsleil-Waututh peoples, and wherever you are in the province listening to this, you are acknowledging elders past, present, and emerging, and our endeavor to ensure that we continually strive for truth and reconciliation. For the purposes of this talk, it's hard for people to ask questions through the Zoom. So if you can put them in the chat, then Brenda can read them out to Matt at the end. For those of you in the audience, you can ask them and then he'll answer them back for the audience to be able to hear. And really, it gives me great pleasure to introduce Dr. Matt Lanktree, who's a clinician scientist and assistant professor and medical director of the McMaster Kidney Genetics Clinic. He looks after people with chronic and end-stage kidney disease at St. Joseph's Healthcare, Hamilton, and McMaster University. And he is a recipient of multiple peer-reviewed funding sources, including the Canadian Institute of Health Research, the Canadian American Societies of Nephrology, and was recently awarded a CSN New Investigator Award. And he really is committed to using genetics to improve the care of people living with or at risk for kidney disease. And so I'm going to hand it over to you and look forward to the conversation.
Thank you, Dr. Levin. Thank you, everyone, for coming this morning on a dark and rainy morning. And so I really appreciate you being here and your interest in nephrology genetics. I should say, as disclosures, I have received speaker and advisory fees from Otsuka, Bayer, Reata, and Sanofi. So my objectives today, this is really a lecture in 3 parts. And so if you're not liking part of it, just wait a few minutes and we'll change gears. So at first, I'm going to tell you about something called clonal hematopoiesis of indeterminate potential, which is a mouthful, but we refer to it as CHIP. And so whether you're familiar with it or not, hopefully you'll get a bit of insight today about what CHIP is and how it might matter for kidney disease. Next, I'm going to talk about the shift to the world of multiomics. So we're moving beyond the GWAS, the Genome-Wide Association Study, to what is emerging as the Proteomewide Association Study, or PWAS. So GWAS, there's TWAS, and now PWAS. And finally, I'm going to talk a little bit about some of our advances in the understanding of the genetics and genomics of polycystic kidney disease. So I start off by showing Galileo's telescope here. And so in 1600, Galileo made this device that increased sight by 20 times and was able to move science away from a geocentric model of the universe. And so my point here is that it was really the development of a new technology that allowed him to look beyond. There was insights, but the technology was a big part of that. And so the advancements in sequencing, genotyping, and biomarker measurements are really the foundation for which these new investigations are able to happen. So first, I'm going to talk about CHIP. And so CHIP is a form of acquired somatic variation. So when most people think of mutations that occur throughout life affecting the DNA, we think of either the loss of heterozygosity of a tumor suppressor gene or the mutation of an oncogene that leads to rapid growth of cells and the growth of a tumor. So cancer, for a long time, has been looking for the presence of these acquired mutations. But what about a mutation that maybe impacts the function of a cell, but it doesn't lead to this rapid growth of the tumor? It doesn't totally take over, but it alters cellular function. Well, we were never really able to identify or measure this based on the limitations of our sequencing technology until quite recently. So clonal hematopoiesis of indeterminate potential, or CHIP, is when there's a mutation within the hematopoietic stem cells within the bones. If these acquired mutations are so deleterious that the daughter cells, the progeny of that cell, do not live, then that lineage dies out. However, if the cell doesn't confers such a reproductive advantage that it takes over and becomes a cancer, then each subsequent daughter of that cell carries that acquired mutation. And so if this is actually pretty common in the population, and again, we've only been able to recognize it with the advances in sequencing technology. So CHIP occurs in these so-called CHIP driver mutations, and it leads to twitchy blood cells that are more likely to send out the cytokines and pro-inflammatory signals that can cause problems in chronic kidney disease. It's prevalent, so about 10 to 20% of people over the age of 65, we can see evidence of these acquired variations. And you can say 10 to 20%, that's a pretty big range. So there's a lot based on how we look and the population that we pick that can change the prevalence. There is an increased risk of forming hematologic malignancies down the road, but it's still relatively low at about 1% per year. And so how do we recognize it? So in next generation sequencing technologies, whether it's exomes or whole genomes, we cut up the genome into these little pieces that are read in parallel on the sequencers. And then we use computers to line those reads up to a reference sequence, and they stack on top of each other. So in this case, the read depth is 24 because there's 24 bars on top of each other. If all of it is the same as a consensus sequence, it's in gray. If it's different than the consensus sequence, that's the colored bars up here. If they're homozygous, so both copies of that gene are mutated, then you get a solid line down because it's changed in every single sequence in that person. If they're heterozygous, you would expect half of them to carry the mutation. Now, before, originally, when our read depths were only in the 20s, we had these occasional things out here, and we're like, oh, these are just artifacts. They must be sequencing errors because they're only in 1 or 2 reads. You can imagine if 10% of cells carry a mutation and they're heterozygous, then only 5% of the reads are going to be that new variant. And so it's only as your read depth gets deep enough that you can statistically significantly say, no, we think this variant is actually there. So together, so Caitlin Vlassert's my PhD student, and Mike Raw at Queens is a hematopathologist who's Caitlin's co-supervisor. And so we started looking at a targeted assay to look at these CHIP driver genes in people with chronic kidney disease. And we had some really interesting pilot data. And then Dr. Levin afforded the opportunity to look at CanPredict, a sub-cohort of CanPredict, to again look at this CHIP and the association with progressive disease. So in this, each point is a person, and the X is their age, and then the colors are people that we identified at least some amount of these CHIP driver mutations. And so 25% of our cohort of people with advanced kidney disease, is the mean eGFR of 27 of lots of different kinds of kidney disease, had these CHIP mutations. So when we looked at the baseline eGFR, both in the Kingston cohort and in CanPredict, we saw statistically significant differences in baseline eGFR. And even more impressively, in follow-up, we found that people were about twice as likely to develop incident 50% decline in eGFR or kidney failure in those that were CHIP carriers. And it was quite consistent between the two cohorts. So we were able to publish this year in JASN. We also looked at other characteristics of chronic kidney disease like anemia. So CHIP carriers were more likely to have lower hemoglobin. They were more likely to have higher doses of ESA, and they had borderline higher levels of some inflammatory markers, but that's still an area of research. So in general, we found about 25% of these people with advanced CKD had CHIP, and it appeared to double your risk of developing kidney failure in the next few years. And as well, they had worse CKD complications. So about the same time our paper was published, there was another cohort that examined people with diabetic kidney disease. And so out of a cohort similar to CanPredict, but a diabetic kidney disease cohort, they adopted a nested case control strategy. So they took 64 people who met the composite outcome of, you can see here, and then they picked out decade-matched controls, 230 controls, and did CHIP sequencing. And they reported 30% of their patients or participants had CHIP, but they came to the conclusion that CHIP was not associated with worse diabetic kidney outcomes. And so we read this with great interest and kind of scratched our head and said, like, what? Was ours like a false positive? Is there some kind of problems with interpretation? Or is it because these patients are diabetic? And curiously, like if people are familiar with APOL1 nephropathy, there's this oddity that it appears that there isn't as an association with progression of diabetic kidney disease with APOL1 because of the competing risk associated with the different. So we said maybe it's just because it's diabetes, but then we delved into the supplemental information, the supplemental, so what the actual mutations that they were calling CHIP were. And according to our filtering criteria, almost half, 51 of 127 variants, we wouldn't have defined as CHIP. And as well, some genetic regions that were covered on our panel were not covered on their panel. And so we said, you know, variant interpretation is hard. So we wrote a letter to the editor talking about how potentially you could come to different conclusions based on how you interpret the variants. So if you include variants as CHIP that are not actually changing the phenotype, then you dilute the signal, you water it down, and you lose that significance. So we said, you know, we were hoping that the authors would reanalyze their data and see if it changed it, but they essentially responded saying, we agree, variant interpretation is difficult. So Caitlin went to Vanderbilt for a research elective with Cassianne Robinson Cohen. And so there, she looked at exomes. So this is a targeted genome panel. So the read depth isn't quite as good as we have with our targeted, but it was based on whole exome data, but they have much larger sample sizes. So 12,000 top med participants. So this was general population. So probably all aware of the explosion of biobanks and population cohorts. And so Caitlin did the CHIP calling in the top med participants and found 9% of people in this general population cohort. Again, age is the strongest predictor of CHIP. So as we age, we acquire these mutations. And so looking at 8 years of follow-up and a primary outcome of 30% decline of EGFR in these people with normal baseline EGFR, again, there was a statistically significant increase in risk of that primary outcome, albeit with a more marginal signal size, effect size. So Caitlin presented this at ASN this year in an oral abstract, and she went on to look at now CHIP in the UK Biobank. So again, this is based on exome data, so it's lower read depth, but again, you can always kind of overcome challenges in measurement with sample size. It was an unfortunate reality. So looking in UK Biobank, an ICD code defines CKD, glomerular disease, again, consistent, more similar in effect size. Again, UK Biobank is a healthy ascertainment bias. The mean age is 50, so lower CHIP prevalences, but again, a consistent direction of effect and really even effect size and statistically significant. Because of the larger sample size, we were able to start drilling down into the different driver genes. And it really appeared that DNMT3A specifically seemed to be different than the rest, that it was less associated with the pro-inflammatory phenotypes. And so if we excluded that gene, the signals actually got stronger. And then Caitlin also looked at the AKI outcomes. So again, these are ICD defined, so they're rough measures of AKI, but again, showed higher risks in both UK Biobank and TopMed of the development of acute kidney injury in patients with, or population members with CHIP. So that's the first portion. So hopefully CHIP you're familiar with, at least now. It appears that there's about a 1.5-fold greater risk of CKD progression or AKI in those who have CHIP. So definitely not like an enormous effect size, but it seems like a risk factor. Heterogeneity exists depending on the gene that carries the variant allele frequency, which I didn't really talk about, but you can imagine there's a dosage effect. So depending on the percentage of your cells that carry that variant, it seems like people with higher variant allele frequencies have more impact. And so future directions that we're working on CHIP include developing mouse models of induced kidney injury and AKI in mice with induced CHIP, as well as looking at additional cohorts with more detailed phenotyping than the population cohorts, such as doing all of CanPredict. And really the question is, could CHIP alter clinical risk stratification or even potentially be a target for therapy? And so maybe in the question period, I can talk a little bit about how it could be a target for therapy. So proteome-wide association studies. So GWAS, was GWAS really that big a deal? And I mean, it found all sorts of loci and like, but is this really important? I don't think is it impacting clinical decision making? So the first one take-home point, or a big, drug targets that are supported by genomic evidence are more than twice as likely to be translated into active therapies. And so drug discovery and target selection and like picking the winners for what should go forward into clinical trials can really be assisted by these type of investigations. So what is Mendelian randomization study? So I was talking about this with the fellows last night, and I don't think any of them had heard of it before. But the underlying concept is that if there's a change in your DNA that alters a risk factor, if that risk factor is in fact causal of the outcome, then the genetic variant, should also be associated with that outcome. And the effect is predictable and testable that genetic variant and that risk factor is, so it supports that the risk factor is causally related. So curiously, I did a Mendelian randomization study looking at LDL cholesterol, say, as a risk factor for CKD progression. So probably everyone knows that the randomized controlled trials of LDL cholesterol reducing were not successful at slowing the progression of chronic kidney disease. And again, Mendelian randomization showed there was no association between genetically predicted LDL cholesterol and CKD. If you look at allopurinol as a treatment for lowering uric acid, whether that would slow progression. So there have been randomized controlled trials that have had weak, haven't been successful. The Mendelian randomization studies, again, so those with genetically higher uric acid are not at higher risk of CKD, which again supports that you targeting a therapy for that isn't going to be helpful. And then blood pressure. So most recently in KI, there was an in-depth investigation that showed that genetic variants associated with higher blood pressure were not associated with chronic kidney disease risk. But in fact, if you looked at genetic variants associated with kidney disease risk, they impacted blood pressure. So but the challenge is that the genetic impact on a phenotype like blood pressure is relatively small. But if we look at biomarkers, the genetic impact on biomarkers, it's much closer. And so there's more variation in the biomarker that's explained by genetics. And so what we said is, could we use this approach to evaluate biomarkers that could potentially be drug targets as impacting chronic kidney disease? So there have been new technologies to measure biomarkers on a high throughput mechanism or high throughput way. So you can measure 1,000 biomarkers in a bunch of patient samples. And so initially people were testing the association between biomarkers and EGFR or kidney disease outcomes. The problem is that kidney function itself impacts the biomarkers. So it's very susceptible to reverse causation. Whereas if we use a Mendelian randomization approach, so we take DNA variants that are associated with lifelong changes in that biomarker, then we can look if that DNA variant is associated with EGFR or kidney disease outcomes to test whether genetically increased biomarker concentrations are associated with kidney outcomes. And so this, if you do it on proteins that are coded from across the genome, this is what in turn is being called a proteome-wide association study. So getting down to the nitty-gritty a little bit here on the X, so each of these black points is a biomarker. Each of these black points is a SNP or a genetic variant, and the effect on the X-axis is the effect of that variant on biomarker concentration, and then the effect on the Y-axis is the effect of that variant on the outcome. So we take multiple variants, combine them, and do this regression analysis to interpret or inter the effect of genetically predicted changes on the biomarker on your outcome. So now on the right side here, this is called a Miami plot. So now each point is a biomarker, as opposed to a variant from the prior GWAS studies, if you're familiar. And so in this case, points that are above the line are those where higher genetically predicted concentrations are associated with the trait, whereas below the line is when lower genetically predicted concentrations are associated with the trait. So we measured 1,000 biomarkers in 12,000 people in a study called the PURE study, Prospective Urban Rural Epidemiologic Study in Hamilton. And I did this with Guy Paré and Salim Youssef in Hamilton, and Nicola is a postdoc who's been working with me. And so we looked at genetically predicted biomarker concentration and its impact on EGFR. So the first one that came right out to the top is Euromodulin, which has been all over if you read the journals, everybody's interested in Euromodulin these days because higher Euromodulin concentrations seem associated with better kidney outcomes. So the only cells that produce Euromodulin are the tubular cells of the kidney. So it's no other cells in the body make it. So more Euromodulin means that you have more tubular cells. And so people in turn have better outcomes. And so people said, could we be increasing people's Euromodulin levels as a therapeutic target? When we look at the genetic evidence, we saw the exact opposite, that genetic variants associated with higher Euromodulin concentrations were associated with lower EGFR. So again, each point here is a genetic variant. You have the regression line. So variants that are associated with higher Euromodulin are associated with lower EGFR and thus an increased risk of CKD. And so we end up with this paradox that, observational evidence is saying that higher Euromodulin must be good, but it's because people have a higher load. They have more tubular interstitial cells, but the genetic evidence is saying that higher Euromodulin is actually damaging. And so higher genetically predicted Euromodulin increases the risk of CKD. And in fact, this is in perfect alignment with what we know from autosomal dominant tubular interstitial kidney disease. So ADTKD is caused by a toxic gain of function mutation in Euromodulin that leads to an accumulation of Euromodulin and subsequent hyperuricemia, gout, and chronic kidney disease. And so it's not truncating mutations that reduce the amount of Euromodulin that lead to disease. It's these toxic gain of function mutations that lead to an accumulation. So again, this follows the results that we see from the PWAS, but it's kind of counterintuitive to what you would expect from the observational studies. So here's Euromodulin off the bottom, and there's even a page break here. But so again, this is a Miami plot. So each point is a biomarker. The horizontal lines here is the Bonferroni corrected significance threshold. And you can see there's lots of biomarkers that are beyond these thresholds where higher genetically predicted concentration is associated with higher EGFR and where higher genetically predicted is associated with lower EGFR. So there's many, so about 6 of these were found in genome-wide association studies. So they serve almost as positive controls of the approach, but there's also some really interesting novel candidates that have not, so we could talk more about those. So next what we did is called a Phenomwide proteomewide association study. And you're like, wow. So now what we looked at is the genetically predicted concentration of the biomarkers on 200 other phenotypes that were available in UK Biobank. And this is important because you can predict side effects of a therapy that altered the concentration of that biomarker. So for instance, looking at Euromodulin again, if you were to genetically reduce, or with a therapy, reduce Euromodulin concentrations, you can see there's a statistically significant increased risk of urinary stones. And so again, we know this from, that Euromodulin helps prevent urinary infections and urinary stone formation. So again, serving as a positive control and looking at some of the other novel targets that came up from the PWAS, so this inhibin beta C, you can see that inhibiting it would potentially increase the risks of, so if you were to investigate this as a potential drug target, you would want to be cognizant of the results from this Phenomwide PWAS. Next, we looked at single cell sequencing. So there's public resources that are able to tell us what genes are expressed in what tissue and cell types. So we said that if the PWAS suggested these biomarkers were causal of kidney disease, then probably that biomarker needs to be made by cells that are in the kidney. And so we were able to do, so this heat map over here shows, so the parts where it's really bold or really dark are, so these are kidney cells that are expressing the biomarkers that came up from our PWAS. And so really what came out, the overall trend is that GFR is not all podocytes and it's not all the filtration membrane. Fibroblasts, interstitial cells, tubular cells are all playing important roles in EGFR and really aren't targeted by our current therapies. So our PWAS identifies novel causal mediators of EGFR. It refines GWAS hits and discovers new signals. And our pathway analysis and single cell analysis really underlies the importance of growth factors and matrix proteins, fibroblasts, and tubules in the regulation of EGFR. And we found some really interesting biologically relevant hits with genetic evidence. So moving away from Mendelian randomization and PWAS to now looking at polycystic kidney disease. So polycystic kidney disease, probably people are familiar, is the most common monogenic cause of kidney failure. It's primarily caused by mutations in either PKD1 or PKD2. So truncating mutations that lead to an absence of protein produced from that copy of the gene are associated with the most severe phenotypes. So here you can see kidney failure on average happens about age 50 in people with PKD1 truncating variants, as compared to PKD2 variants. Average age of kidney failure is in the mid-70s. So in general, we can see there is a phenotype-genotype correlation. But it's important to remember that those are the averages, and there's still a distribution of outcomes regardless of mutation type. So people with the most severe PKD1 truncating mutations can still have age of kidney failure ranging from mid-30s all the way to mid-70s, which really, is important when we're counseling patients that this is not deterministic, right? So there are still things we can do to improve your care, to improve your outcomes, to delay kidney failure. So when we investigators all around the world really have taken patients that did not have PKD1 and PKD2 mutations and then did exome testing to look for other genes that could be mutated leading to cystic phenotypes. And so here's PKD1 and PKD2 in the nucleus. After they're transcribed, they move out to the endoplasmic reticulum where they get translated, and then they get modified and transported through the Golgi apparatus and all the way out to where they reside in their in a complex PC1 and PC2 joined together. And people are still arguing about the exact function of this PC1, PC2 complex on the cilia, but there are multiple proteins that are involved in the maturation and transport of this PC1, PC2 complex. And what do you know? It turns out that many of the genes that are involved in this trafficking pathway are those that, if they're mutated, can lead to a reduction in the dosage of functional PC1, PC2, and they lead to cystic phenotypes. So cumulatively, so you may have heard of GNAB or DNA JB11, ALG89, FT140. HNF1 beta is kind of a separate disease, but is associated with cysts in the kidney as well. Sex61 and 63 usually have more. So actually, many of these have more liver phenotypes than kidney phenotypes. So they can have both liver and kidney cysts. Some of them are also associated with diabetes, like HNF1 beta. So cumulatively, there's a list. So it's not like, oh, what's PKD3? I mean, we now know there's 3, 4, 5, 6, 7, 8, 9, 10. There's about at least nine additional monogenic causes of kidney cysts. And the next, so cumulatively, they account for maybe 1 in 20 ADTKD cases. So I think IFT140 is probably going to turn out to be the most common of these additional causes, but it's still less than 5% of all comers with cysts in the kidney. As a whole, they have relatively slower kidney disease progression. They're all milder than PKD1 and especially milder than PKD1 truncating mutations. They often do not have classical phenotypes. So whether it's the distribution, the progression, the total kidney volume growth, that's pathognomonic of PKD. And really, it's yet to be seen whether these people need to be treated differently, whether they're going to be responsive to the same therapies or not. We don't really know the answer to that. Another gene that's kind of come out, or group of genes, is the Col4A, so type 4 collagen. So I hope everyone here is familiar with what I call type 4 collagen nephropathy, which used to be called Alport syndrome. So you say Alport, and people think about young boys with hematuria, hearing loss, and vision abnormalities that have progressive chronic kidney disease. But we now recognize that type 4 collagen nephropathy is a much broader presentation that includes that Alport syndrome, but also includes women who are hemizygous. So one of the Col4A5 is on the X chromosome, so one of their 2 copies, or heterozygous cases where people used to be called benign familial hematuria, but we know that these people are at increased risk of proteinuria and progressive chronic kidney disease, and the cardiovascular disease that comes with that, and in some cases even kidney failure, especially in the context of diabetes. There are also important causes of FSGS, but there have been case reports and even case series of people identifying type 4 collagen variants in people with cysts in their kidney. But the question when you come back is these aren't entirely rare in the population. A couple percent of the population carry these variants. So is it just a coincidence that you're sequencing them because they have cysts and then they have these variants of uncertain significance in the type 4 collagen genes, or are they actually causing the cysts? So I presented this at ASN this year. And so in Dr. Pei, my former postdoc supervisor's cohort in Toronto, we looked at the prevalence of type 4 collagen variants in people that had a known PKD1 or PKD2 mutation versus those that did not have a PKD1 or PKD2 mutation. And so you would expect if they were just a passenger, if they were just there by chance, you would see equal prevalence of these variants in those with genetically defined and not genetically defined. What we actually saw was about a 10-fold higher rate of type 4 collagen variants in people who did not have a PKD1 or PKD2, suggesting that there's at least statistical evidence that there's an enrichment of these variants in people with cysts. We next went to look at the phenotypes of the people that carried these. So you can see they predominantly had what are classified as atypical, so fewer larger cysts, unilateral presentations. They did not, as a whole, have kidney enlargement, so they had lower Mayo Clinic classifications. And if you look at the rate of eGFR decline in these people, you can see there are a couple that have advanced CKD, but as a whole, most of these people with kidney cysts and type 4 collagen variants had maintained kidney function. So changing gears a little bit, who are the people that I think should get genetic testing in ADTKD? And so this is, I think the pendulum has been swinging back and forth with regard to this question. Initially, people said, and my next slide, MRI is the best assessment of risk progression. So it's for determining risk of future disease. You have a lifetime of progression up to that point. And clearly people that have big kidneys that are completely full of cysts are at higher risk than those that have only a few and smaller kidneys. So MRI is the best risk assessment tool we have. But the real role for genetic testing is diagnosis in cases that are somewhat atypical. So people that don't have a family history, people that we really want to know, so donor assessments of family members, and we want to be sure that they don't have the familial variant. Pardon me. Situations where there's early or severe presentations, where there's discordance in disease severity between family members, discordance between the rate of kidney function loss and the total kidney volume increase, because I said that's pathognomonic for PKD. And if there's other features that make us think that there's like a syndromic form, like nephronephrosis. So, obesity, supernumerary digits, recurrent lung infections, developmental abnormalities, all those things that make us think like maybe there's something more here than just PKD. And finally, I put here atypical imaging because, PKD should be, if it's germline, it should be in every cell in our body. It should be affecting both kidneys equally. And so these atypical imaging, what is their risk of progression is not clear. So moving on to atypical PKD. So we looked at again, the Toronto cohort and looked at the rate of progression of kidney failure in people, or of kidney disease in people with atypical imaging characteristics. So here you can see this person has one large cyst on the right kidney, but otherwise has pretty normal looking renal parenchyma on the right-hand side. This is a different MRI weighting. So here the cysts are white. And so even though there's a significant cyst pattern here, the left kidney is totally normal, and there's still significant portions of the right kidney. So it shouldn't be totally surprising that these people have better outcomes. Importantly, the Mayo Clinic classification paper by Razabel in 2015, they upfront excluded everybody that had atypical imaging. So there wasn't really anything in the literature confirming that these people have slower progression. So we recently published this in scientific reports. These people have atypical. Now, the other kind of interesting thought is that there's a much higher rate of no mutation detected in these patients. And so there's kind of two possibilities. One is that it's not PKD1 or PKD2. So do they have like a CACUT, a congenital abnormality of the kidney and urinary tract phenotype, or do they have somatic mutations? So I talked right at the beginning about somatic variants in our blood cell lines causing CHIP, but what about somatic variants in the cells in our kidney? So if people have either a mutation throughout their development or growth throughout life, it's possible that some of their cells have acquired the mutation that's leading to that deficiency of PC1, PC2 that leads to the cystic phenotype in part of the kidney. But if we do genetic testing in circulating blood, that mutation isn't there. And so our genetic testing is negative. So certainly the challenge, these people aren't biopsied. You would really need to look at cadaver specimens to try to figure out if indeed there is a clonal expansion of a somatic variant in these people with atypical imaging. So I've flown through and talked at a mile a minute, so hopefully you found it interesting. I talked about first the somatic acquired variants leading to clonal hematopoiesis, or what we call CHIP, which is a risk factor for AKI and CKD, but I should say that it's really turning out to be a risk factor for almost every chronic disease. So it started really looking at coronary disease. It's a risk factor, a similar 2 to 3-fold increased risk of coronary artery disease and myocardial infarction. It's a risk factor for non-alcoholic steatohepatitis and fatty liver disease. Really across the spectrum, inflammatory diseases like arthritis. So, and even some interesting stuff looking at frailty and cognitive outcomes as well. So CHIP is really an exploding area and in need, for further study and working out the mechanisms and potentially treatments to lower the risk associated with it. PWAS, the Proteomewide Association study, I imagine that's probably the first time maybe some of you ever have heard of this term, but the future is multi-omic. So it's going to be including single cell sequencing data. So I didn't present a TWAS or any TWAS data here, but transcriptome-wide association studies is another in this multi-omic space. And then including the GWAS and the exome sequencing as well. And finally, there's a wide range of genes now, at least 8 or 10 on top of PKD1, PKD2 that can lead to multiple cysts. Generally, they have milder phenotypes, which is good from the clinical perspective, that most people wouldn't confuse these as high-risk patients. But certainly, as the cost and availability for genetic testing improves, I think people need to become aware of these because they are going to be coming across, and it can really help with the both the risk in children and providing the genetic counseling to the patients and understanding why they have cysts. So I think it's still And I think that pendulum is going to swing from only providing genetic testing in the cases where it's going to impact clinical management to potentially swinging towards where it provides information and a diagnosis, and that's enough. So with that, I'd love, there's lots of time for discussion, and thank you everyone for coming this morning.
Questions, turn on your microphone so that everyone else can hear. So stimulating conversation, and
I have here my mentors and previous supervisors and my students and my funding.
Great. Questions or thoughts that people have? Nichols, sorry, this was your thing here.
I have to hold it? Okay. That was complicated. Not the talk. The talk was very straightforward and clear. So what I wanted to say was, it seems like CHIP is an acquired mutation, and you were kind of making that point in Mendelian randomization, that if you have a genetic variant and then you develop like a phenotype or a risk factor, then you can conclude that they're related. But if it's an acquired mutation, can't you still be kind of stuck in that fallacy of chicken and the egg? Because they can develop diabetes, and then unless you check at a second time point, like that they have that variant.
Fantastic question. Yeah.
Congrats. Do you do multiple analyses?
Well, you're bang on. You're absolutely right. So, and that's a really impressive question. Great insight. How do we know that the chip is causing these outcomes and not that these outcomes are? So, I mean, the first kind of thing, the fact that it's associated with lots of different badness still even goes into, is it an effect or is it actually driving the association with these? So beautiful. So one tool we could use to look at that, and look at that, I love the fact that you put together the part 1 and part 2. It's a really impressive question. So can we use Mendelian randomization? So the first part is, in order to do Mendelian randomization, you need to have a genetic instrument that predicts the risk factor. So you could do a genome-wide association study of the likelihood of developing CHIP. So certainly, I'd say this is ongoing. There have been a couple inferior studies that have published with this kind of approach. This is something that, so if we can find genetic predictors of the risk of developing CHIP, then those should also be associated with the outcomes. And that would suggest that CHIP is actually causal. So that's one way of getting at it. Another way of potentially getting at it is the model organisms. So if you can cause CHIP and then end up with phenotypes, that's another way that you can infer causality. So another kind of way that you could look at it is
Emergency Department Trauma 1. Code blue, Emergency Department Trauma 1. Code blue, Emergency Department Trauma 1.
So another kind of thing, like if CHIP is actually causing kidney disease, you would expect the so it's really in the macrophages, and that are the pro-inflammatory macrophages. And so, we should see evidence of those CHIP carrying macrophages within the kidney. So can we take biopsy samples and show that those macrophages that are carrying these variants are actually infiltrating the organs that get affected by CHIP? So again, this is an interesting idea and something that there's efforts to make. The challenge is that, only a couple % of the macrophages carry that variant. So even if you do like flow or something to try to pick out those macrophages to see if they're the ones that are actually carrying it, it's not straightforward. But certainly, again, great question, and it's something that people are working on, trying to figure out ways to do that. Another potential way is to lessen the impact of CHIP, and if that prevents the outcome. So is there a therapy that we can do? So I said these twitchy macrophages that are carrying this variant are sending out all these pro-inflammatory signals that are causing problem. If we give a therapy that blocks those pro-inflammatory signals, is that a therapy for people with CHIP? So there was a trial recently called the CANTOS trial, which used something called canakinumab, which is a monoclonal blocker of interleukin-1 beta. I'm certainly not an expert in interleukin-1 beta or this trial, but they recruited people with elevated CRP and it was secondary prevention of coronary disease and treated them with this canakinumab. And so it had, not landslide benefits, but then in a post hoc analysis, they did the sequencing of the CHIP, and it turns out people who had CHIP, the effect of the drug was much better than just people who had generalized elevated CRP. So again, the next question is, could we recruit a population of people with CHIP and then redo that study with that? And again, so whether it's coronary disease or it's kidney disease, but great question and something that we're working on.
The chat there. Go ahead.
Okay.
So summarize that question. Are there any environmental factors that aggravate the link to CHIP? And can SGLT2 inhibitors be beneficial as it decreases mitochondrial dysfunction?
So, first question, risk factors, biggest smoke, I gave away the answer. Biggest environmental risk factor is smoking, which again, isn't that surprising. So there's a huge association. So most of our analyses, if we have smoking data, we adjust for smoking because smoking is strongly associated with the development of, and I already said age. I mean, age is the biggest risk factor for everything. And so again, part of the challenge is even if you try to adjust for age, it's probably often inadequately adjusted for the impact of age because age is associated with everything bad. And if age is associated with CHIP, then all of a sudden CHIP's associated with everything, even if you try to adjust for the age of the participants. So certainly I would suspect that any exposure to radiation, or who knows, even the flight I took here, increases your risk of developing acquired variants. So certainly there are environmental associations with CHIP as well. The second question was, so it's SGLT2 and mitochondrial function. So it's an interesting, I'm not really aware of the link between SGLT2 and mitochondria. So I am going to have to look into that. Certainly we are looking at the impact of CHIP on, so mitochondrial function is another big phenotype in aging. And so I've been asking and wondering if there could be a relationship between CHIP and mitochondrial function, but again, that's in its early days of investigation. And I think that's also a pretty exploding area of research. Whether SGLT2, I have no idea whether it would impact CHIP. I mean, it seems like SGLT is good for everyone. So except for maybe PKD, but that's a bit of an inflammatory comment.
Thanks, Matt. That was great. My question's particularly in the PKD population. So I'm really interested in the idea that, of course, there are so many more genes than PKD1 and PKD2 that underlie all of this. We all have a few patients that have significant liver burden compared and out of proportion to their kidney burden. And I'm intrigued by your statement that it is those patients that may in fact have, genes outside of PKD1 and PKD2. Have you found that? Has that played out clinically? And do you see a role for almost routinely providing those sort of outside of PKD1 and PKD2 gene panels to those patients?
So I should add there's lots of PKD1 patients, and it seems like PKD2 actually has a more severe liver phenotype than PKD1 for reasons that aren't known.
Yeah.
Usually really bad liver disease with less severe kidney disease, the first thing I think is PKD2, and that's going to be the most common cause of it more often than not. It can be, so for reasons that aren't understood, it seems more common in women than men to have significant liver involvement. And so people talk about estrogen hypotheses and association with pregnancies and oral contraceptive pills. The other thing is that the growth of the liver seems to taper off after menopause. So I don't know if it's been your clinical experience, but in mine, it's mostly women in their mid-40s that have the most trouble with the huge livers. And the good news is that it often tapers off. I do co-manage some of these people with 2 hepatologists that have been using the somatostatin analogs to try to prevent the growth of the liver. It doesn't impact kidney progression, but these people usually have relatively milder kidney disease. So like GNAB and DNA-JB11 both have liver cysts. They're not usually like really bad. The really bad ones are still PKD2 most of the time, or else they have ADPLD, which is another autosomal dominant polycystic liver disease, is another disease altogether that does not have kidney cysts, but has liver cysts. And so like Sex63 is one of those kind of crossover genes. And it can be totally debilitating. And it's really frustrating and hard to treat these patients because we don't have great therapies. And they need a liver transplant often, partial hepatectomies. If they end up with sclerotherapy or draining procedures, they get refractory ascites. It can be really, really devastating.
Yeah, as a follow-up, so in our clinic here, I mostly solitary manage these patients with significant liver
There's no
involvement.
hepatologist you can convince.
Yeah, and I've sent a few now for liver transplant assessment, and a few have gone on to receive liver transplant. And as you said, it's very debilitating because there's not much to do.
And their liver function still works.
Absolutely.
And so they're like, oh, their MELD score, forget about it. And
Yeah.
they're like pregnant with their huge liver. It's terrible.
Yeah.
But their liver still works. And so then.
Yeah. Thank you.
Yeah. And yeah.
Again, yeah, thanks. Fantastic talk, Matt. To your comment of, maybe the pendulum swinging back with genetics, one of the things I've thought, especially when you see and some of your work with kindred that have very, very different outcomes, as we learn more and more of these both other genes and kind of the accessory genes downstream, any planned work in the future, do you think some kind of role of, you could almost envision a multigenic kind of risk score, right? Where you're not just looking at the one causative gene, but actually putting together a profile that might lend to a higher risk, especially as we try earlier and earlier in children to prognosticate them when they won't have those imaging changes yet.
So we were talking about last night at dinner how you can have 2 children that are so different.
Yes.
And it's fascinating to me. So like if you have 2 family members, 2 brothers and sisters, siblings that have the same main effect mutation, they both have polycystic kidney disease, but then they're only half their genetic is the same. They have that variant the same, but they still have on average half. It's possible they have much less than that in common with their family member. So, Peter Harris at Mayo Clinic started a genetic modifier study that looked at genome-wide association studies in people that had, so initially they tried recruiting people with similar main effect mutations to try to overcome the heterogeneity that's associated with the main mutation type. They're having challenges with it. So if there isn't an easy answer, genome-wide genotyping in people with polycystic kidney disease has not been done very well across, which is part of the reason why I want to get more patients together, because I completely agree with you that polygenic risk for diabetes, polygenic risk for obesity I don't see how it possibly could not be associated with risk of progression. So, but still, it's not published, no one's shown it, but I would wager a lot.
Just a corollary question to the notion that you can predict response, that you can target drugs, but you could also probably predict responsiveness and unresponsiveness to drugs.
Yeah.
Theoretically, and it would be really interesting because for all that we give one drug to one disease, perhaps there are people who will not benefit, and you would look for a different drug. Or
so clinically, and probably you guys can tell me your experiences, but one of the challenging things about using tolvaptan, which is the only approved cyst reduction or cyst modifying therapy in polycystic kidney disease, is that, the natural history of PKD is to lose between 3, 4, 5, 6, maybe 7 mils per year of VEGFR, but it's not like tolvaptan fixes that. their GFR still goes down. And so the patient comes and is like, how do you know this is working? My kidneys are still getting worse. And I have all these symptoms and all this blood work and all these problems associated with it. My kidneys are still getting worse. Like, is it even worth it? And so I have to say, like, oh, well, in the big studies where we took 1,000 people and we put in the people who were on it were slower. So you just got to trust me. But they're like, well, how do you know that I'm not a non-responder because I'm still getting worse anyways? And they say, oh, can't you re-image me and show that my kidneys aren't growing as much as it can? But the new ERA EDTA guidance says do not do serial imaging because there's enough variability in TKV estimation that serial MRI is not a good measurement of response to therapy. People say, oh, why don't we use like urinary concentration? And then originally people said, oh, well, if their urine osmolality is 150, then clearly we're blocking ADH and let's call it a day. But now people are saying, well, probably we shouldn't be targeting our dosing to a spot urine osmolality because it's a 24-hour drug. And in the trials, everybody was on maximum tolerated dose. So we should be pushing people to max dose. So again, this is a bit of a, it's frustrating that we don't have a biomarker that's reliable enough of response to therapy to say to people, look, it's working. You're not getting worse, because people are still getting worse. They're just getting worse slower. But that's a hard thing to convince people of.
No, it's interesting also just to go back to the beginning with CHIP, because if you think about air pollution and all the things that are environmental exposures, and there's higher kidney disease and lung disease in places that are highly polluted, like there may well be some.
Well, and that's just hematopoietic.
Right.
But I mean, what about other cells in our body? And this is really, again, it's the new instrument. It's the new magnifying glass that we're able to start measuring this acquired variation that does not lead to the growth of a huge tumor. It was not long ago, I said this last night too, that I was like, oh, I like genetics because we can take a sample and it's the same for the rest of your life and it's the same in every cell in your body. And now people are like, oh, we need to do serial DNA collections. Before, all these population biobanks, they did baseline DNA and that was it. They said, we have the DNA of everybody. Now we're like, oh no, we have to go back and recollect on everybody because maybe their DNA is changing.
Yeah, we used to say it doesn't matter when you do it in the study, it'll always be the same. It's a
Yeah.
constant and it's not constant. Yeah, no, yeah. Anyways, it is 8:30 and it is a Friday, but I want to thank you tremendously for the time you spent with the fellows last night and also for this excellent talk and really stimulating lots of great thought and collaborations across the country and internationally. So thank you so much for coming.
Thank you, everyone. Thank you.